Alright, so in this video, I'm going to be running you through the topic of gluconeogenesis. Alright, now gluconeogenesis is simply a pathway that the body undergoes so the main aim is actually to produce glucose. Alright, and we know that glucose at the end of the day, we need to produce ATP. Okay, now when you look at this process, some tissues such as the brain, we have the brain, the red blood cells, so the RBC cells, the kidney, the medulla, and so on and so forth. Alright, so you find that they need a continuous supply of glucose as a metabolic fuel. Alright, so in cases where liver glycogen, okay, which is a very essential source of glucose, where it cannot meet the needs for the brain, the RBCs, the kidney, okay, looking at the normal fact that it's able to provide energy for about 10 to 18 hours. Okay, this is the amount of time that the liver glycogen is able to supply the body with actually glucose so that energy is produced. Okay, now when you look at this time is limited. All right, when we look at a scenario where all the liver glycogen stores are depleted, the body looks for other means and ways to actually provide glucose, and in the end provide energy. So the brain actually uses up 70% of glucose, okay, that's produced during normal by the liver during normal fasting. So 70% of glucose used by the brain, okay, so used by the brain comes from the liver. All right, so this is to tell you that if the brain is able to use 70% of the glucose that comes from the liver, whenever the liver glycogen is depleted, it means it now has to rely on gluconeogenesis, which is the process that we're going to be talking about quite shortly. All right, so when you look at gluconeogenic precursors, all right, before we even go further, we're going to look at gluconeogenic precursors. Okay, so these precursors, they can be intermediates that are found in other cycles. For instance, the glycolysis and the citric acid cycle, okay, as well as the lactate cycle or Cori cycle, glycerol, and so on and so forth. So you're going to see familiar components that we're going to talk about that are coming from the citric acid cycle, the glycolysis, and so on and so forth. All right, so these are like shuttles. If an element is missing in the gluconeogenic pathway, it's able to come from the citric acid cycle or glycolysis in order to supplement for that. All right, so basically, when you look at gluconeogenesis, it's like the reverse of glycolysis. So gluconeogenesis is the reverse of glycolysis. Okay, so having said that it's the reverse of glycolysis, there's going to be a series of reactions that are going to help us, okay, in order for you to know that this is the reverse, because glycolysis has three chemical reactions that are irreversible. Okay, then the other 7 reactions are actually reversible. Okay, so you're going to note this. So when you look at, we're going to look at a briefing. When you look at the lactate, okay, lactate is an end product of when pyruvate is worked on, okay, and this lactate can also give us the chemical element pyruvate that I earlier on mentioned. Pyruvate is what is being used in the process of gluconeogenesis. Alright, so we have a mix-up. So another intermediate that I had talked about is oxaloacetate. Okay, oxaloacetate. Now this oxaloacetate, I think I'll highlight it. Remember, oxaloacetate is coming from the citric acid cycle or tricarboxylic acid cycle. Okay, that's where oxalatate is coming from. We also have a protein that we call alanine. Alanine can also give you pyruvate. Okay, so these are the pathways that we're going to be talking about. All right, okay, so the formation of glucose does not occur by a simple reversal of glycolysis. So remember, in glycolysis you first start with glucose all the way to pyruvate. Okay, now when you talk about, so let me write this as glycolysis. Okay, then in red, gluconeogenesis goes the reverse way. Okay, so meaning it's supposed to come from pyruvate and give us back glucose, whilst glycolysis goes from glucose going to pyruvate. However, the reversal is not that easy. We do not just jump from one reaction to the other. Remember that there are 3 chemical reactions that are irreversible. So those are the ones that actually have to be altered so that we're able to get back to glucose. Alright, so instead the glucose is synthesized by a special pathway that we are calling right here, which is the gluconeogenesis. So it requires that both the mitochondria, so we have 2 organelles, so the mitochondria as well as the cytoplasm are all supposed to work. Alright, they all work. They have special enzymes that actually work. So during an overnight fast, okay, there are things that you're going to notice. During an overnight fast, approximately 90%, okay, 90% of gluconeogenesis occurs in the liver. So it occurs in the liver. Okay, so during an overnight fast, what you're going to notice is 90% of gluconeogenesis will happen in the liver. Okay, with 10% being in the kidneys. 10% in kidneys. Okay, of the newly synthesized glucose molecules. Alright, but however, if at all that fast prolongs, okay, the kidneys become a major source of a major glucose source of glucose production. Okay, so what you're going to notice is that during a fast, 90% of this gluconeogenesis will happen in the liver, but when it's prolonged, the kidneys are the ones that actually take it up. Alright, so we're also just going to do a brief recap on glycolysis so that you're just able to see how we are able to build up from there. Okay, so remember, in glycolysis we said it starts from glucose. Okay, the next it goes when glucose is now phosphorylated, so it becomes glucose 6-phosphate. Whenever from glucose 6-phosphate we now have fructose 6-phosphate. Okay, from fructose 6-phosphate we have, it's broken down. Now before it's broken down, it's converted to fructose 1,6 bisphosphate. Okay, from fructose 1,6 bisphosphate, it's broken down to two elements: dihydroxyacetone, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. Okay, so this is just a brief step of glycolysis. From here we know what's next. You have 1,3 bisphosphoglycerate phosphoglycerate. Okay, from here you have 3-phosphoglycerate. Okay, from 3-phosphoglycerate you have 2-phosphoglycerate. Okay, then on you have phosphoenolpyruvate. So I'll just put them in initials, which is PEP. Then the last thing that you're going to have is finally the pyruvate. Okay, so gluconeogenesis main aim is to come from pyruvate all the way back to glucose, surpassing all these reactions. Okay, now however, like I said, we have 3 irreversible reactions that need to circumvent, meaning they need to find their way around, okay, in order for this whole process to go back to glucose. Alright, one of them is reaction number 1, where we have hexokinase being the enzyme. Then we also have where we have PFK1, okay, and where we have pyruvate kinase. Okay, so reaction 1, 3, and 10, these are the reactions that are actually irreversible. So even when we start looking at glycolysis, you will notice that immediately we get to pyruvate, we immediately skip all these steps, okay, because these are reversible. We can easily go back to the previous. We'll skip all these steps, and when we come to the steps that are irreversible, this is where we'll talk about special reactions that need to occur so that glycolysis is just able to take place. Alright, so the first thing that we're going to talk about is what exactly happens to the pyruvate after it's formed via glycolysis. Alright, so the first reaction that occurs is carboxylation of pyruvate. Okay, you can write this down. So first thing that happens is carboxylation of pyruvate. This is the first thing that happens in the first reaction. So what you're going to notice is that pyruvate, okay, pyruvate combines with a bicarbonate, okay, plus the help of adenosine triphosphate. Okay, so pyruvate is converted to oxaloacetate. Okay, oxaloacetate plus, so this is on the product side, ADP and an inorganic phosphate. Remember that we are ripping out this phosphate from ATP and giving it to pyruvate. Okay, and we are also adding carbon dioxide to this. That's why it's called carboxylation. So what you need to note is that the enzyme that's used in the first reaction is called pyruvate carboxylase. Pyruvate carboxylase. Okay, so this pyruvate carboxylase is what actually catalyzes the process. Okay, now another thing we need to note is that there is a coenzyme. All right, so this coenzyme is like a companion to pyruvate carboxylase. It's actually a vitamin that we call biotin. Vitamin biotin. So biotin also helps in the carboxylation of pyruvate to oxaloacetate. Alright, so you may be asking yourself, why is pyruvate being converted to oxaloacetate? Okay, so we're going to answer that question very soon. Alright, so now what you need to note, another important thing is that pyruvate carboxylase is allosterically activated by acetyl-CoA. So we can simply write it as is activated by acetyl-CoA. Okay? So acetyl-CoA actually activates pyruvate carboxylase. However, it's inhibited by ADP. Okay, ADP. So ADP is an inhibitor, whilst acetyl-CoA is an activator. Alright, so oxaloacetate. So before we go further, you need to notice that these reactions are happening within the mitochondria. Okay, so if this is the mitochondria, okay, so this is your mitochondria. What you're going to notice is that, so when I write OAA, this is oxaloacetate. So oxaloacetate has to be converted to phosphoenolpyruvate. So oxaloacetate needs to be converted to PEP. Okay, in order for gluconeogenesis to continue. So just take you back to this. So oxaloacetate has to be converted to PEP. Alright, so what you notice is that if this is your OAA, you know that pyruvate cannot directly go back to PEP. Okay, because this reaction, remember, is irreversible. So what does the body do? It converts the pyruvate to a chemical that can easily go back to PEP. Okay, that's why we said these chemicals are able to circumvent, or rather find their way around, so that they're just able to reverse the reaction. That's why oxaloacetate has to be converted to PEP for gluconeogenesis to continue. So the enzyme that catalyzes the conversion is actually found in both the mitochondria and the cytoplasm. Alright, so we can find it in the mitochondria as well as the cytoplasm. Okay, so these are the organelles that we're able to find these enzymes. So the phosphoenolpyruvate that is generated in the mitochondria. Okay, so I'll just draw a diagram right here. This is your mitochondria, this is your PEP. Okay, the phosphoenolpyruvate that is generated in the mitochondria, like you can see, it has to be transported to the cytosol by a specific transporter. Alright, so remember, the membrane has got transporters that are able to transport these chemicals out with ease. So it needs a special transporter to take it to the cytoplasm. Whereas so this can go out and come in the cytosol. So this is your cytosol, this is your mitochondria. Okay, whereas the phosphoenolpyruvate that's in the cytosol, okay, before we go to that, you find that the phosphoenolpyruvate, in order for it to form in the cytosol, okay, it requires that the transport of oxaloacetate from the mitochondria into the cytosol. Alright, so there are two procedures that we're going to talk about. Alright, just right about now. So point 1 is oxaloacetate is unable to directly cross the membrane. So if this is oxaloacetate, remember it was formed through carboxylation, it cannot go outside in its form. Okay, it needs to be converted to something else so that it's able to go out of the mitochondria. Alright, so what happens is that it has to be reduced to malate. So OAA must be converted to malate. Okay, and when it's converted to malate has the ability now to go out of the membrane. It's able to be transported from the mitochondria and into the cytosol, where it's reoxidized to oxaloacetate. So when malate goes out of the cytosol, okay, it has to be reoxidized back to oxaloacetate, and then oxaloacetate will now be converted to PEP. Alright, so this is the procedure, and remember that the enzyme is called malate dehydrogenase. Okay, so this is what happens. So let me just draw a diagrammatic diagram that you're going to see, illustration, sorry. So you have your malate right here, right? This malate, this is your cytosol. Okay, malate can be converted back to oxaloacetate. Alright, remember this is our membrane, and this is what we call the malate transporter. Okay, and then this is your mitochondria. So mitochondria, what you're going to notice is that oxaloacetate is being converted to malate. Alright, now there is reduction that's happening here. Okay, and in reduction there's a chemical that we call NAD+. It's converted to NADH. Alright, because it's removing a hydride. Okay, so there's reduction that's being used. There's a chemical that's going to gain a hydrogen, and in this case, like we mentioned, the enzyme is called malate dehydrogenase. Okay, so this is the procedure. From the mitochondria, oxaloacetate cannot transfer itself to the other side. Okay, this is our oxaloacetate. What does it do? It's converted to malate. Malate has a special transporter across the membrane. Alright, when malate is able to go through, malate is immediately reoxidized. Okay, meaning that that hydrogen that was removed, that was taken, is now brought back. Alright, we have NADH. Alright, so what happens is that there's reoxidization and oxaloacetate is formed in the cytosol. Okay, so this is what happens. Next thing that we're going to talk about is decarboxylation. Decarboxylation of cytosolic oxaloacetate, cytosolic OAA. Alright, it's a very important step. So remember that we had undergone oxidation reaction okay, converting the malate to oxaloacetate. Now, once the oxaloacetate is now in the cytosol, it has to be decarboxylated, meaning that that carbon dioxide that was added has to be removed. Okay, this time we're going to use guanosine triphosphate. Okay, so once it's decarboxylated, it's then converted to phosphoenolpyruvate, okay, plus GDP. Okay, it's going to be guanine diphosphate, because remember the inorganic phosphate was removed and the carbon dioxide has now been retrieved. Okay, once we now have our chemical phosphoenolpyruvate that you're able to see, okay, it means it's going to be the phosphoenolpyruvate is acted on by reactions from the glycolysis. So you're going to notice this. Okay, so we're going to look at those processes that we had written down in glycolysis. So once oxaloacetate is turned back to phosphoenolpyruvate, it's going to undergo a series of steps. So all these reactions are going to occur until it becomes fructose 1,6 bisphosphate. Okay, so it skips to this, and remember that this process is irreversible. Okay, that's why it cannot go all the way to glucose, because it's an irreversible step. All right, so once it undergoes those glycolysis reactions, as it is running in reverse, there's something we're going to actually notice. All right, what we're going to notice is that the PEPCK is actually inhibited. So don't worry about the initials, these are actually enzymes. Alright, the phosphofructokinase, phosphoenolpyruvate carboxylase kinase. So the PEPCK is inhibited, so it's inhibited by ADP. Okay, so this is an inhibitor. So this enzyme is the one that's driving the reaction from OAA to phosphoenolpyruvate. This is the enzyme. Now what they're saying is that ADP inhibits this whole reaction. Okay, causes it to go slower. Another step that we're going to talk about, so this is just a point to take note. Another step that we're going to talk about is dephosphorylation of fructose 1,6 bisphosphate. So I'll take you back to the glycolysis pathways reaction. So once we had fructose 1,6 bisphosphate, we're now here. Okay, this is the process that we are at. Okay, what we are going to see is quite interesting. So what happens now? Fructose 1,6 bisphosphate undergoes dephosphorylation. Okay, meaning a phosphate has to be removed. Remember, we have fructose at carbon number 1 and carbon number 6. Now for us to have fructose 6-phosphate, which is the intermediate fructose 6-phosphate, it means the phosphate on carbon number 1 has to be removed. That's why it's called dephosphorylation. So fructose 1,6 bisphosphate is hydrolyzed. Okay, water is added to it to break that bond. Okay, in order to give us fructose 1,6 bisphosphate plus that phosphate that was on carbon number 1. All right, So what you notice is that this reaction is an important regulatory site. Okay, so take note, this is a regulatory site of gluconeogenesis, so meaning you're able to regulate the process, okay, through this step. So this is a regulatory site. So what you notice is that this fructose 6-phosphate, it has inhibitors and it also has activators. All right, so we're going to mention that quite shortly. So fructose 1,6 bisphosphatase is the enzyme. Okay, so I'll just highlight it as enzymes involved. Okay, so these are the enzymes that are involved. The fructose 1,6 bisphosphatase. Okay, that's the enzyme that's involved. So it is inhibited by one, elevated levels of AMP. Okay, so AMP inhibits this enzyme. All right, now let me just take you back to, we'll keep on referring to glycolysis. So remember that for us to move from fructose 6-phosphate to fructose 1,6 bisphosphate, sorry, the enzymes that are involved. Now in the step that we are at as at now, they're telling us that actually in high levels of AMP are able to inhibit this enzyme fructose 1,6 bisphosphatase. All right, and then while high levels of ATP and citrate stimulate gluconeogenesis. So I want you to just link whatever you are learning in glycolysis. What you notice is that during glycolysis, ATP and citrate are inhibitors. Okay, they inhibit those processes. But when you look at gluconeogenesis, they're actually stimulating, or rather they are supporting gluconeogenesis. This is to tell you that these two pathways cannot happen at the same time. All right, so fructose 1,6 bisphosphatase that's found in the liver and the kidneys can also be inhibited by fructose 2,6 bisphosphate. So fructose 2,6 bisphosphate. Okay, all right, let's just write that nicely. Bisphosphate. So this is an enzyme that is able to just inhibit. All right, so you find that that's why it's called a regulatory site, because you can determine whether glycolysis will happen or not and whether gluconeogenesis will happen or not right at this site where we have fructose 1,6 bisphosphate. All right, so this is what exactly is happening. So after the whole process occurs, you're going to notice that now immediately we have fructose 6-phosphate. We're now going to go to dephosphorylation of glucose 6-phosphate. So remember, from fructose 6-phosphate, according to glycolysis. Once we add this, fructose 6-phosphate can easily go back to glucose 6-phosphate. However, glucose 6-phosphate cannot go back to glucose because this process is irreversible. So what happens? Hydrolysis has to occur again so that we are able to remove that phosphate from glucose 6-phosphate, and so that we can just have glucose. Okay, so from fructose 6-phosphate we are now at glucose 6-phosphate. Okay, now this glucose 6-phosphate needs to undergo hydrolysis. Okay, meaning water is being used in this case. So you're going to see this equation where we have glucose 6-phosphate, okay, being added onto water to give us the glucose that we actually want, plus the inorganic phosphate group. Alright, so this is our final thing that we're going to have. Now what you need to notice is that release of the free glucose requires two proteins. Okay, release of this glucose that has been made, that you're able to see over here, it requires two proteins that we call glucose 6-phosphate translocase. So I'll just write it as G6P, meaning glucose 6-phosphate. So glucose 6-phosphate translocase needs to transport glucose 6-phosphate across the endoplasmic reticular membrane, okay, and then the enzyme that's in the endoplasmic reticular membrane, the glucose 6-phosphatase, removes the phosphate, producing the free glucose. Alright, that's what's going to be happening. So I'll just draw something short. Okay, so what you notice is that you have your G6P right here, right? Now there's an enzyme, glucose 6-phosphatase, so glucose 6-phosphatase, okay, I'll just write tase at the end, catalyzes reaction from glucose 6-phosphate to glucose, and thereby releasing a free phosphate group. Okay, now we're saying that this glucose cannot just go out of the endoplasmic reticulum. It needs glucose 6-phosphate translocase. Alright, so, and remember that glucose has these GLUT channels. Okay, so once this comes out, it's now able to come out. If this is the kidney or whatnot, we have what we call GLUT channel. So this is GLUT2. Glucose can go out through this channel and into the capillary. Okay, goes into the capillary and then on. These are red blood cells. So once it goes in the capillary, it can now be used as a source of energy. Alright, this is how glucose is obtained from the gluconeogenic pathway. Alright, I hope this is clear. The next thing we're going to talk about is hormonal regulation of gluconeogenesis. So we're talking about the hormones that are involved. Hormones involved. Okay. So the hormones involved in this case, we're going to talk about glucagon, and we also have insulin. We may mention epinephrine here and there, but these are our main point of reference. Alright, so when you look at glucagon, okay, so basically we're just going to look at what's the function of glucagon. So this is our glucagon. So remember, whenever your body runs out of whenever it runs out of energy, or rather glucagon is the enzyme that enables the liver glycogen to be released, okay, so that we have enough glucose in the body. Whereas insulin, when there is too much glucose in the body, it facilitates the storing up of insulin of glucose in the body through glycogen. So what you notice that is glucagon, number one, it lowers the levels of fructose 2,6 bisphosphatase. Bisphosphate, sorry. Okay. So this is what is actually happening. Alright. So it lowers the level of fructose 2,6 bisphosphate. Now remember that this fructose 2,6 bisphosphate was an inhibitor. Okay? So if it was an inhibitor in gluconeogenesis, meaning the inhibitor has now been blocked, and therefore gluconeogenesis can be favored in this case. Alright? Because remember, this fructose 2,6 is the one which inhibits fructose 1,6 bisphosphate. Okay? Now, if this fructose 2,6 bisphosphate has been inhibited, it means fructose 1,6 can now function and it will now favor gluconeogenesis. Okay, this is the reaction that's going to be favored. Another thing that you're going to notice about glucagon is it elevates CAMP levels, cyclic AMP. Okay, and it also has to do with the CAMP dependent protein kinase activity. Okay, so I will not write that, I'll just mention it. So the CAMP dependent kinase activity is the one which stimulates the conversion of pyruvate kinase to its inactive phosphorylated form. Okay, so we have pyruvate kinase, right? We have pyruvate kinase, and remember, pyruvate kinase is converting phosphoenolpyruvate to pyruvate. Alright, now what's basically being said is that CAMP is the one which stimulates the conversion of pyruvate kinase, which we're able to see here, right, to its inactive phosphorylated form. Alright, so this means that immediately, remember, we have oxaloacetate here, right, in the process of gluconeogenesis. Immediately oxaloacetate is converted to phosphoenolpyruvate. What CAMP does is that it inactivates this enzyme pyruvate kinase, because pyruvate kinase loves to convert phosphoenolpyruvate to pyruvate. Okay, so when oxaloacetate is converted to PEP, this CAMP goes to act on the enzyme that's able to convert the PEP back to pyruvate, so that gluconeogenesis can continue and not be disturbed by glycolysis. So in short, the whole point of these enzymes is to inhibit enzymes that are favoring glycolysis, okay, and rather stimulate the enzymes that are favoring gluconeogenesis. So the enzymes that favor glycolysis are inhibited, whilst the enzymes that favor gluconeogenesis, they are not inhibited, they're actually activated. Alright, so glucagon can increase the transcription of PEP carboxykinase gene, increasing the levels of the enzyme. All right. So insulin, when you look at insulin, it increases the activity of PFK1, okay, phosphofructokinase 1. All right, pyruvate kinase and levels of fructose 2,6. So this is when we're talking about insulin. Okay, so insulin will increase the activity of PFK1. Okay, remember PFK1 is an enzyme that's found along the process of glycolysis, which I'll show in a minute. Okay, all right, so remember we have phosphofructokinase 1 somewhere here. Okay, so we have the enzyme. If this is our enzyme, right, or rather if our enzyme is here, phosphofructokinase 1 is converting this to this guy or this to this guy. Okay, this is just an example. So it's that enzyme is stimulated so that the reversal reaction can occur quite quickly. All right, so pyruvate kinase and the levels of phosphofructokinase fructose 2,6 bisphosphate are increased, and I'm sure you remember how we talked about fructose 2,6 activity. All right, the next thing that we're going to talk about briefly is just the Cori cycle. So the Cori cycle is like a subset of gluconeogenesis. Okay, so we have the Cori cycle. So what happens in this Cori cycle is that where we have gluconeogenesis, right? So we have gluconeogenesis right here, and we know that glucose is being produced from pyruvate. Okay, and pyruvate can come from lactate. Remember that image that was drawn in the first place? So this is the blood. Okay, and this is glycolysis. Glycolysis. So in here we have glucose. It's the other way around. Glucose is being converted to pyruvate. Pyruvate to lactate. So this is just simply to tell you that it's a cycle. From glycolysis, we're able to have pyruvate. Okay, from pyruvate it goes to lactate. Lactate goes out in the blood. Okay, this is in the muscle, by the way. This is muscle, and then this is the liver. Now I want you to notice a scenario where this is a runner, right? And whenever a runner is, they're in sports or any other sport, what you notice is that usually the body has less time to produce ATP via aerobic glycolysis. So what happens is that this is going to be anaerobic, meaning no oxygen. Okay, so no oxygen is used. So in this case, when pyruvate is made, it's converted to lactate, and in some cases lactic acid that people are, even when you are running, you're able to feel it in your knees. It hurts a lot whenever it accumulates. Now this lactate, whenever it's produced in the muscles, right, it goes out, goes in the blood, then the lactate goes in the liver where it's converted to pyruvate. When pyruvate undergoes gluconeogenesis, and that is what gives it glucose. Alright, so this is the Cori cycle. Okay, the Cori cycle is just simply telling you what happens when someone is coming from glycolysis and it's going to gluconeogenesis. Alright, So thank you so much. That's all for this video. I hope you enjoyed it.